Hi Year 7. Today we're going to be looking at the different parts of a microscope, but first we need knowledge organisers, paper and a pen. So please make sure you have all three before we start. Okay, so once you've got your knowledge organiser and your paper and your pen, pause the video, okay, and attempt these retrieval practice questions about plant and animal cells. So pause the video and have a go at these. Okay, so for question number 1, the 3 structures found only in plant cells are the cell wall, chloroplasts, and the vacuole. So if you got all those right, give them a tick. And then for question 2, we have the nucleus, cytoplasm, cell membrane, and mitochondria that are found in both plant and animal cells. Okay. So for this lesson, the LO is to know the different parts of a microscope, and the success criteria are to be able to label the different parts of a microscope and be able to set up a specimen. So, using your knowledge organiser, pause the video and have a go at labelling this diagram of a microscope. So use your knowledge organiser, pause the video and see if you can complete these 4 labels on the microscope. Okay, so for the one at the top, we should have the eyepiece lens. The one on the left should be the stage. Then we have on the right, in the middle, the focusing knob. And then finally, we have the objective lens. So remember, there are 2 lenses on a light microscope. Okay, so if we were asked to make a slide, which is what we would put under a microscope to allow us to view a cell, we would have to first get a slide, which is a thin piece of glass, and it's used to hold objects which are examined under a microscope. Most slides will already be made up for you. If you get the chance to prepare your own slide, here's what you would do. So if we were using onion cells, we would cut open an onion. We would use forceps, which are like sharp tweezers, to pull a thin layer of skin from the inside. We would then lay that layer of skin, which is called the epidermis, on a microscope slide. We would then add a drop of iodine solution to the layer, and we would carefully place a cover slip over the layer. We would carefully place this so there were no air bubbles trapped underneath the cover slip. Now, iodine solution, iodine is a chemical stain. We use it to make it easier to see the parts of the cell. This is what onion cells would look like under a microscope. So what I want you to do is pause the video and see how many of the organelles or structures that we mentioned at the start of the video you can see here. See how many you can label, okay? See how many you can identify. So pause the video and have a go. Okay, so we should have here around the very outside of the cell, we have the cell wall. Okay, remember an onion is part of a plant, so these are plant cells. So we have the cell wall. Just on the inside of the cell wall, the inside line of the cell wall is the cell membrane. We can see the cytoplasm here, okay, and we can also see the nucleus. We can't see any other organelles very clearly on this image. So we should have cell wall, the membrane, the cytoplasm, and the nucleus. So, I said before that a microscope has two different lenses. So it has the eyepiece lens and we have the objective lens too. These both magnify the image that we will be looking at. So here we have the equation to work out the total magnification of a light microscope. We would multiply the magnification of the eyepiece lens by the magnification of the objective lens. So using the equation that is on the screen, have a go at calculating the total magnification. So for these examples, the eyepiece lens will always be times 10. So eyepiece lens magnification in the equation at the top here will be 10. The objective lens magnification changes each time. So all you need to do is make sure you have 10 here because that is our eyepiece lens magnification. You need to times it by these three numbers to get three different answers for the total magnification of the microscope. So pause the video and have a go. Okay, so for the first one, we should have 10 times 4. So remember that eyepiece lens for these questions is always times 10. So we put 10 into our equation. We then have times by 4. So we multiply it by the objective lens magnification. That is here, the times 4. So 10 times 4 is 40. That means that the total magnification of this microscope is times 40. For the second one, we have 10 again as the eyepiece lens magnification, like it says in the question. And this time, the magnification of the objective lens is times 10. So the sum we're going to do this time is 10 multiplied by 10, which should give you 100. That means that the total magnification of this microscope with the eyepiece of 10 and the objective piece of 10 should be times 100. Okay, so the objective lens is times 10 and the eyepiece lens is also times 10. So that gives us 100 and that means the magnification is times 100. Now for the last one, we have again the eyepiece lens magnification of 10. This time, the objective lens magnification is 40. So our sum is going to be 10, which is the eyepiece lens magnification, times 40, which is the objective lens magnification. That gives us 400. So the total magnification of the microscope is times 400. Okay, so these are the questions we need to be able to answer. So what I want you to do is pause the video, have a go at these questions. So the first one is to label the parts of the microscope. We have already done that, so have a go at doing it yourself. Then you need to check your spellings, okay? When I put the answers up, what we need to do is make sure that we've spelt all the different parts of the microscope correctly. If we haven't spelt it right, then we need to write it out 3 times. Then we need to use our own words to explain how to set up a specimen of onion cell to observe through a microscope. Then we explain why iodine is used to prepare a slide. And then we have some more magnification questions. So pause the video, have a go at these, and then I'll go through the answers for you. Okay, so the first one, we should have labelled the eyepiece lens, the focusing knob, the objective lens, and the stage. Please check your spellings of these. If you spelt any of these incorrectly, please write them out 3 times with the correct spelling. This will help you learn how to spell these words. C. In your own words, explain how to set up a specimen of onion cell to observe through a microscope. I've put on this slide the method that I showed you earlier. If you have similar steps to these 5 steps, if you've covered all these steps in your method, then that is fine. It doesn't have to be exactly these words, but we need to make sure that we are cutting open an onion. We're using forceps to peel the thin layer of skin from the inside. We're laying that onto a microscope slide. We're adding iodine, which remember is important, so we can see the cell clearly. And then we're placing a cover slip over that layer. Iodine is important because it is a chemical stain. Okay, we use it to stain different parts of the cell to make it easier to see them. E is another magnification question. This time the eyepiece lens is 20 and the objective lens is 40. So if we put that into our equation, we have eyepiece at 20 and objective at 40. So 20 times 40 equals 800. So this means that the total magnification of the microscope is times 800. F. What would the magnification be if the eyepiece was 50 and the objective lens was 250? So we do the same again. We put the eyepiece magnification into the equation, which is 50. We're then going to times that by the objective lens magnification, which is 250. 50 times 250 gives us an answer of 12,500. So the total magnification of this microscope is times 12,500. The final question asks you to calculate magnification if the eyepiece lens was 40 and the objective lens was 150. So we do the same again. We use our equation. We find the eyepiece lens magnification, which is 40. So we're going to do 40 multiplied by the objective lens magnification, which is 150. So our sum this time is 40 multiplied by 150. That gives us 6,000. So our total magnification of this microscope is times 6,000.